Good evening to all the participants. So, welcome to the 18th session on bioinformatics approaches for drug discovery. So, coming into applications. So, we'll be discussing about various methods of drug design in this session. So, before going to this session, can we recap the last session contents? What did we discuss in the last session? So, kindly recap the last session topics. What exactly did we discuss? So we also discussed something related to applications, isn't it? Very good. So Keetana has answered string, funcoop. That is, these are protein-protein interaction databases. Just we started from that. Excellent. Okay, so I think one participant is providing answers for the entire participants team. Huh? So even others can try. Good. So we just started with string funcoop, and then we came up with some basic concepts of synthetic biology. And for using the synthetic biology tools, that is, if you want to build compartments to form a genetic circuit, okay, using a models to develop a genetic circuit, we need synthetic biology open language. So that we have discussed, and I showed you the website and URL. And hybridization, we discussed a part of a process for DNA microarrays. Then we discussed what for DNA microarrays are used. I told you that it's used for gene expression. If it is a protein microarray, protein-protein interaction can be determined, including protein binding sites and mRNA decay rates. Okay, and the life, everything we can determine uh, these kind of applications using microarrays. We have seen also types of microarrays like spotted DNA array, oligonucleotide chip, etc. And we have seen the types of clustering techniques, agglomeration, hierarchical, nearest neighbor, complete, and I think one or two. Okay, so it's something is left. But okay, we have discussed about the types of clustering techniques. Do you know what is exactly clustering? When we have to discuss about these types of clustering, these clustering, we see if you get multiple datas in the microarray. I told you in a microarray, you may have 10,000 spots to 20,000 spots. And if you find these data, okay, and if you put in the, and if you check the data, okay, how you link one data with the other data. Okay, so this is something related to clustering of data. We see the types of clustering. Okay, so through the clustering, we can able to identify after doing the statistical analysis, we can normalize the data, and we come up with a percentage of gene expression. And through the percentage of gene expression, we can tell what is the exact stage of cancer if you are diagnosing for a cancer. Okay, good. Excellent try from a participant, Keertana. Very good. Keep it up. And now let me take you to the bioinformatics approaches for drug discovery with no further delay, because we have so many concepts to cover. I see that I can, yeah, I can take the take. I try to cover concepts fast, but if you are not following, message me or message in the Google Meet, or you just unmute and just ask me to slow down. Okay, because I have prepared almost 51 slides. Um, now so just I start with this first. How researchers discover the drug for a disease? Okay, so different scientists are there working in different areas of engineering. How this is generally discovered, etc. So for this, first we understand some drug discovery applications. You know, if you see the drug development in the market, okay, so initially we do the computational screening. Okay, the computational, after the computational studies, we go for preclinical, we go for after computational studies, that is target discovery and screening, we go for lead development, and after that we go for preclinical development. Okay, and after the preclinical only we go for phase 1, phase 2, phase 3, phase 4. So these are the phases happening in the pharmaceutical drug development. Okay, So earlier it has taken almost 1 to 3 years for computational. Here, if you see, they have mentioned up to 5 years. Up to 5 years they have taken for computational experiments. Now, is it happening like that? No. Now, even if you see this pandemic situation, discovering a drug for SARS-CoV-2, okay, most of the researchers, scientists all over the world, even the top scientists in USA, they have gone for repurposing of drugs. Okay, some scientists taken already existing viral drugs and then just they tried which is best for SARS-CoV-2 strain. Some scientists have taken all the molecules from a large databases like Zinc. In there is a database called Zinc 15. In Zinc 15, you have uh, billions of molecules. Okay, nearly 2.7 billion molecules. I remember. So he has gone for virtual screening. So plenty of molecules are there, billions of molecules are there. A scientist from California, he is in his lab, is having a supercomputer. He tried screening all these molecules. Actually, if you see, zinc contains over 230 million purchasable compounds and 3D formats, and zinc also contain over 750 million purchasable. So it comes around 1 billion compounds in zinc 15 alone. Okay. So he has taken all the compounds and he just gone for virtual screening with the spike glycoprotein. Virtual screening is one of a molecular docking method, but instead of screening a single drug, you're screening multiple drugs with a particular target receptor, with a diseased receptor. That is, in our case, if you go for SARS-CoV-2 infection, if you want to get a drug, we use a spike glycoprotein. Okay, so here if you see, there is one option called tranches. So if you click the tranches, okay, we have 2D and 3D. If you click this 3D, you get only the 3D structure of molecules. Generally, if you want to go for docking, I generally suggest you to take the 3D. Okay, even when I'm explaining you about NCBA PubChem, you remember, I told you to only select 3D and download STF. Okay, if and if you select STF, then you can download in that kind of data set. And this data set can be visualized using Discovery Studio Visualizer. Okay, so first I am showing you a kind of a database called Chain. Here you have more than 1 billion molecules. And if you see here in the Chink branch, okay, this is a kind of a table with multi dimensions. For example, in molecular weight, it ranges from 200 to 500 from left to right. And log partition coefficient, okay, the log p values start from -1 to 5 or greater than 5. Okay, if you want to select a molecule of with a particular physical property, it can be like 350 is the ideal molecular weight, and if you want to select log p as 3, okay, then 350 and log 3, you can see 37 million compounds are present. If you if you click this, okay, that 37 million molecules will be downloaded in the form of a single data set. So all these 37 million molecules, and if you go to with 350 with 2.5 log partition coefficient, you find here log partition coefficient. So you find 48 million molecules. Okay, so based on a particular physical properties, like if you see true digital electrophoresis, the proteins are separated based on molecular weight and isoelectric point. Similarly, even these compounds are categorized based on molecular weight in column wise and log P in row wise. So, but if you select this, okay, all these molecules, okay, can be downloaded. Okay, can be downloaded. So just you have to select it and you have to click here. Then you can go with so these kind of a screening you can't do with your laptops or normal desktops. You should have a high configuration systems to check which is the best compound out of all 37 million compounds. Okay, so you got it. So there are so many protocols for drug design discovery. So this is one such protocol. Okay, taking go to the database, take millions of molecules and just screen which is the best one. So if you screen and get a best one, then the best drug what we identify can be a drug or active pharmaceutical ingredient. The best you identify is the lead. So whatever lead you identified, you go for development. in lab, you try to generate it. If it is a synthetic molecule, do the synthesis and develop the molecule. Okay, so the computational, according to this, they're telling it takes 4 years, but when the world has ended with a pandemic, okay, declared as a pandemic in March 2019, a scientist immediately in California has downloaded all these molecules, not even is specific to the molecular weight and log partition coefficient. He downloaded all the billion molecules and just came up with a best compound from the virtual screening, and he published the same in May 2019. So it means that he has taken just not more than a month to go for virtual screening. So now the computational process is rapid. Okay, so we can even if you go with some other new infections, viral infections, we can able to complete computational process. Okay, not only discovering a small molecule. If you see the SARS-CoV-2, we generally go with mRNA, isn't it? So even if you want to get a kind of an mRNA, if you want to get a kind of an antibodies, okay, it may take extra 1 month or 2 months. That's it. Now, computational process, we can able to complete maximum of 3 months, even discovering a macromolecule for a particular disease target. So, so we can complete as soon as possible. Then we can go for lead development, and then we can test with preclinical. You, if you see how when the drug came to the market, okay. So almost it has taken nearly 1 year, 1.5 year during that pandemic time. And during that 1.5 year, like from March 2019 to September or October 2020, the many clinicians prescribed the antiviral drugs like anti-dengue, anti-malaria, anti-chikungunya. I mean, sorry, these kind of a drugs. Okay, so whatever drugs used for treatment of these diseases, these drugs they supplied as a repurposing drugs for SARS-CoV-2. Whether it works, at least I told you some these three, four drugs. Out of 8 drugs, there is one more drug called anti-HIV drug. Even anti-HIV drug is prescribed by the clinicians all over the India. It happened, okay, but those who got infected earlier, even they have taken this kind of anti-HIV. Is that anti-HIV? I mean, HIV is linked something with SARS-CoV-2? No, isn't it? It's not integrated with genome at all. Only the SARS-CoV-2 spike glycoprotein can interact with the ACE protein, angiotensin converting enzyme protein. That's it. But you can't tell that a SARS-CoV-2 is integrated with our human genome. So it happened, but even the scientists got confused and they don't know what exactly they can provide a solution. And so many things happened with the repurposing drugs. Okay. But only if you check with animal studies, okay, whatever drugs can be small molecule to the macromolecules, and then check with healthy volunteers in phase 1 and phase 2, where phase 1 you go with just 100 healthy volunteers, phase 2 with 1,000 healthy volunteers, and from phase 3 onwards you check with disease volunteers. If only if the drug is safe in the healthy volunteers, then we can go in checking the efficacy, bioequivalence, bioavailability of drug in the disease patients, which happens in phase 3 and phase 4 clinical trials. Okay, and obviously, even if it gives some kind of a side effects after the patient's taking for a year, okay, we may get ill with some side effects. Then that kind of a toxicity is estimated, and even if you have a new drug which can replace the drug with a nil side effect, this will be stopped, and obviously this will be popularly spread in the market. Of course, if you go with the if you see the last phase, it takes you to the post marketing surveillance. But in post marketing, if it surveils, if your drug surveils without any side effects, no problem. If it fails, then obviously what drug can clear the phase 4 will end up with more marketing. Okay, and if you see this kind of application areas for modeling and simulation, there are different areas like protein structure prediction. You remember this is most important. If your structure is not present, for the spike glycoprotein, scientists all over the world face difficulties in getting a 3D structure from X-ray, NMR. So they find very difficult because it's more infectious and it's the type, the class type pathogen is, I think there's a maximum type of pathogen which can get infected just through air. So if so, because it's an airborne pathogens, isn't it? So they have tried with cryo-electron microscope. That's the best technique they adopted. So meanwhile, the scientists try to use a protein structure modeled from SARS-CoV-1 spike glycoprotein. Before SARS-CoV-2, we also had a SARS-CoV-2 strain. I think in 2010 it came to India. Okay, so we had a structures for that. So using that they modeled, and from that I think even that scientist, the California scientist, has docked billion molecules with that modeled protein, because it has taken some time. They didn't get immediately in March 2019. Only few years, I mean, few months after few months, I think they get a kind of a best resolution protein, and then with even some kind of a precise kind of drugs also came. Repurposing drugs came after the discovery of the protein structure. So, protein structure prediction is one of the important phase in drug discovery, homology discovery. Like if you see the FDA approved drug for treatment of SARS-CoV-2 is remdesivir. Remdesivir. This is the only FDA approved drug up to present. The remdesivir generally targets the polymerase enzymes. Okay, but this is the only drug up to present. And so uh, this for this, if you want to get another any other drugs with a FDA approved, so one can able to check the remdesivir related drugs, whether any related drugs can give a better efficacy. Okay, so we can start with the FDA approved, and we can end up with more similar drugs, and which can show you a better binding results with the target receptor. And pharmacokinetic modeling, function prediction. I told you pharmacokinetic modeling in MATLAB, symbiology. Function prediction. This is a function annotation, like going with the motive finding. You remember, scan pro site can gives you the motive, can be a structural motive or functional motive. Similarly, if you go for MEME, still the function can be predicted with the motive. Okay, a part of a sequence can give you a function. Process modeling, toxicology prediction. So many areas are there, including disease simulation. Now, let us take you to the traditional practice. And earlier, we use only the pestle and mortar, and whatever products you derive from the natural can be plants, animal sources. Okay, generally, these they use for treatment of various diseases. So, mostly we go with a herbal, isn't it? So we synthesize many compounds with a single plant. For example, if you want to treat viral infections, then better we choose neem. That is the best plant, best tree available in our country, isn't it? Neem is famous for treating any viral infections. Okay, so if so, if you just grind the neem leaves in the pestle mortar, and generally we can give for the treatment of viral infections. But whether you know all the compounds present in the neem leaves can give same antiviral treatment, can able to relieve from the viral infections? No, we can't tell. If you have some 100 compounds, okay, I'm telling the most important 100 compounds, may some 70 compounds give significant results, and the 30 compounds may not give the significant result. It may even suppress the viral inhibitors. Okay, it may. So then we have to find which is the best compound which is giving an antiviral activity for a specific virus. So this can't be done with the traditional drug design. That is, this kind of a drug design happened 40 years before. Nowadays we are looking after a particular drug, not all the drugs, whatever you take, even from the neem leaves. So you take only the specific drug, one or two base compounds. You take it and you try to talk with a particular target of a virus. Then only you can come to know. So one should have a knowledge of 3D structure of both ligand. Ligand here is a drug, a compound, inhibitor, agonist. Whatever are the terms I used, this term for a ligand is generally a small molecules. This can also be large molecules like proteins, protein antibodies, DNA molecules. Even the DNA can also be used. Okay, I mean in the form of a DNA dependent DNA polymerase, something like that. Okay, so generally, if you see this, I'm sorry, DNA can also be used. Okay, but DNA dependent DNA polymerase is a target receptor. So target here I mean generally diseased proteins. Target is disease protein, ligand is the drug. Simply you can remember like this. If only if we have a 3D structure of a drug and a diseased protein, then only we can able to come up with a structure-based drug design. This is more precise, and even you can take less dosage when compared to the large dosage and number of doses. Okay, so and if you see the to relieve from the cancer, that is, if you go for a chemotherapeutic treatment for treatment of cancer, one has to reduce the dosage level and number of dosages. Okay, that we should keep it in mind. And if you see this kind of a medicine, okay, Zolgensma, okay, this data I taken 8 years before. So at that time itself, the price of this single short medicine is around 18 crore. This is manufactured by the Novartis. Only one company is producing this kind of an medicine. Okay, and this is generally using to treat spinal muscular atrophy. The children during the after the birth, that is during 1.5 years, 1.5 years, the child suddenly can develop this kind of a disease, spinal muscular atrophy, and that means he lose. That the child will be losing contacts with the brain. So they behave like a mental patients. It happens suddenly. Up to 1.5 years, you don't find any symptoms. But this has happened just because of genetic disease, genetic hereditary genes. Some genes can cause a spinal muscular atrophy. So only you have to go with a gene targeted delivery for this disease. Okay, in a particular age, it starts from 1.5 age to 6 age it seems. Okay, up to the age 6, anyone can develop. I mean, not anyone. One in a million will be getting this kind of a disease. One in a million, that is one in 10 lakh people. If it happens, then obviously one has to go for treatment. Only one, they are going to treat. They are going to target this kind of a human SMN protein. Okay, so for this, they have developed a vaccine, but the success rate is not 100%. Even if you take a single shot, you can't tell that they will be getting relieved. Sometimes it leads to fatal death within 2 to 3 days. If they are surviving, they may get relieving soon. It depends. Okay, this of course either if you want to cure, you have to take this drug, and many people go for applying funding and so now we understand the importance of drug discovery. A single shot, 18 crores. How about a paracetamol? So most of the people suffer fever, isn't it? Normal fever. But think about the price. Just 1, 2 rupees. That's it, isn't it? Based on the brand. Okay. Now let us take you to the computational methods in drug design. So there are various methods of drug design. Okay. The most first used phase is checking the physical properties of the drug molecules. After checking the physical properties, we can go for drug likeness prediction. Okay, predicting the drug likeness, we have so many filters, drug likeness filters, and we have already chem informatics softwares like MolSoft and Mol Inspiration to predict the drug likeness. So not only we can just check the drug likeness using the MolSoft and Mol Inspiration, even we can predict from the databases like NCBI PubChem. In NCBI PubChem, we have the properties of physical properties and chemical properties of the drugs. Using the properties, one can able to tell the drug likeness. So other than that, we can also use these team informatics software. Then I can tell you what's the difference between the database and these softwares. Now let me take you to the next ADMET prediction. That is absorption, distribution, metabolism, excretion, and toxicity. So this is something related to pharmacokinetics. So if one has to determine the pharmacokinetic properties of a drug, one can use the tools like Swiss ADM and pre-ADMAT. And you remember, so whatever the techniques I showed and the tools I showed, most of the tools are online tools, open source softwares. Anyone can able to practice, except some softwares like this software. This Discovery Studio QSR. This is a commercial one. This is a commercial one, and of course DS Ligand Fit. DS refers to the Discovery Studio. We have a commercial version in our institution, so I just mentioned, but this is a commercial one. So and also molecular dynamics NAMD. Where is it? Somewhere. Okay, but Chromax is the free one. You can try with the Chromax. So the other programs are freewares. Okay. So for admit prediction, you can go with the Swiss ADME pre-admit, QSR, QSPR, go with Galaxy. Uh, Galaxy is a free online tool. Pharmacophore modeling, Pharmagist, PharmaMapper, molecular modeling, Swiss Molar ITSF. I think we have taken extensive sessions on molecular modeling of proteins, molecular docking, autodock, inverse docking. That is just a reverse docking. We also call it as a reverse docking. We use a server called ACID. It's a free one. Okay. Molecular dynamics, Chromax. So these are some of the approaches. And we also have a mini project discussion, isn't it? So in mini project discussion, I can show you some 6 different topics. Okay, that I'll be discussing it on tomorrow. So I can give you some brief ideas, but whatever you select, okay, whatever you select most, I can brief you like how I can able to proceed because all I have taken nearly 6 mini projects. I can't able to guide all the 6 mini projects at for a 2 sessions. You need a you have to select based on the selection of projects by the participants. Like if many participants have selected drug discovery, I'll be taking you to the projects to guide the projects on drug discovery. If many are taking something related to genomics, then I have to guide on genomics. Okay, this is how we can make it out. But how to work, I can give you a kind of a brief description. Okay, so that I'll be giving it on tomorrow along with the cellular computing, that is DNA computing. Then on next week, okay, so I give you sufficient time because Saturday and Sunday you look through whatever I'm going to deliver it on tomorrow, okay, and then you can ask me, okay, you can give me a choice to focus much on which kind of a projects. Then I can take you accordingly and I can complete a kind of a small project, okay, a small project demo in this online. You got it? So that we see out on tomorrow. So everyone try to attend. Don't miss my classes. Each and every class you feel valuable at the last time because you're connecting all the computational skills in the form of a mini project to acquire uh, to apply in your curriculum or to apply where you can get a remedy even for industrial solutions. For industry, we have to provide the solutions, isn't it? And even for a society, healthcare, we have to provide solutions. Of course, this is a very important informatics called drug discovery in bioinformatics. So, virtual screening methods to discover the drug for a disease. There are so many methods. This is one of the popular method. Virtual screening, if you have plenty of drugs, even if you see in NEEM, if you have 100 important compounds for treatment of viral infections, okay, which is the most potent viral compound, which is going to treat a specific disease like chikungunya? And what is the specific compound in NEEM which is going to cure chickenpox? Okay, so even the drugs are there which can give the remedy to a particular virus. Then you just have to isolate a particular compound. Don't just take an entire name and you can tell I got a drug for that. Okay, it's a kind of an Ayurveda. We have Ayurveda, Siddha, Unani, homeopathy. There are different branches of medicine, isn't it? Other than allopathy. So if your doctor is prescribing a paracetamol and you are approaching accordingly, a clinician, a pediatrician, then you're going for an allopathy type of medicine. But we also have other branches. So if you take neem leaves, it's a kind of an Ayurveda, Chorna, from 2 or 3 plants. Again, you're going with this. Okay, so you're going for either Ayurveda or if now you are coming to the Siddha. Okay, so it depends, but better try whatever we try in practice in general. We can try with even a specific drug comes under an allopathy medicine. Flowchart of ritual screening, 3D structure of target. Okay, if you have that is target, I mean a disease protein. If you have, uh, you can able to go with that is if only if you have a 3D structure of a ligand and then 3D structure of a target, then you can go with structure-based methods. The best example is molecular docking, protein ligand docking. Okay, but if you don't have this structure, you have this structure that is a drug structure alone, then better go for ligand-based methods. The ligand-based methods, if actives are known, you can go for similarity searching or pharmacophore mapping. And if you have actives and inactives, better go for machine learning methods. Got it. If actives are not known, if you don't have an active pharmaceutical ingredient, they just it's a kind of mimic compound of existing drug, then we call it as an inactives, yet to be synthesized, and you have to check the properties of it. So that we can able to use for that we can use machine learning methods, artificial intelligence. Got it. So don't use AI in machine learning just like that. If you already have a database and softwares. Types of virtual screening methods in drug discovery. Ligand-based generally go with pharmacophore modeling QSAR. We will be seeing the subsequent slides about all these computational methods. Molecular docking and MD simulation, you go for structure-based. Hybrid, either ligand-based or structure-based. Okay, that is a best example is the pharmacophore-based docking approaches. So it uses both the principles, either ligand-based or even the structure-based. Okay, that is pharmacophore modeling or pharmacophore-based docking. What is molecular docking? From the beginning, I was explaining about docking. I told you there are majorly two technical concepts. I mean, majorly two technical terms. One is ligand, one is ligand. The other is the receptor. The receptor is usually a large one. Okay, in our case, that is in this case, enzyme is your receptor and ligand is your substrate. Now think about this kind of an applications. Okay, so wherever you find a molecular recognition, okay, what we speak in molecular biology. So, in terms of biological events like transcription, translation, signal transduction, transport regulation, enzymatic catalysis, viral and bacterial infection, and immune response, all these things in computationally we term this as molecular docking. See, if you see this antigen antibody interaction, where can we find this? This we can find in immunology, in immune response, isn't it? In immune response, the antigen antibody interaction is gives you what antibody is. Antibody is generally a kind of a drug, a macromolecular drug, which generally fits with the pathogenic antigen. The antigen is derived from the pathogen source. If it is a cancer, it can be a cancer antigen if it is a cancer. And if it is a viral thing, then a bacterial antigen protein is your. But in our case, generally in this immune response, most probably the antibody is your ligand. The antibody is small size. The antigen is large in size. They are showing only the small one, but if you see the antigen, it is a very large one. But only a part of a surface receptor is binding with the antibody. Exact term for antibody, even if you to see in cancer treatment, the antibody is what you give it as a drug. So this is a ligand. The antigen is a disease protein, which is a receptor. So when compared to this, you can follow that. But here in this enzymology, the enzyme is your receptor, which is a large macromolecular structure. Substrate is what the substrate. You have already studied the enzymology. That is enzyme concentration plus substrate concentration can give enzyme substrate complex. Then this enzyme substrate complex in the reaction can give you enzyme and product, isn't it? This is the reaction we follow. And here if you see, this is a signal transduction. In signal transduction, okay, the molecule, for this I can give an example like hemoglobin. Hemoglobin carries oxygen as the molecule. If oxygen is carried by the hemoglobin is nothing but blood protein, main blood protein. So this main blood protein is a transport protein because it's going to carry oxygen. During inhalation, the oxygen is carried out by the hemoglobin, then it forms a oxyhemoglobin. Okay, that we call that as a oxyhemoglobin. That is a different which from a hemoglobin. But during exhalation, carbon dioxide is going to release from the hemoglobin. then at that time we call that as a carboxyhemoglobin. So in that case, we have a different protein form that is carboxyhemoglobin. So oxyhemoglobin, carboxyhemoglobin, if carbon dioxide is carried out. Okay. So obviously what you understand, it is a transport process happening on. Okay. Transport regulation can be done based on the inhalation exhalation. And obviously here in this case, we can find a signal transduction. This is a signal transduction. The best example I can provide you is again immune response. That is generally IL-2 ligand bind with the IL-2 receptor. GM-CSF ligand bind with the GM-CSF receptor. IL-1 ligand bind with the IL-1 receptor. So generally you have a ligand and receptor in interleukins. I mean interleukins. If you have studied immunology, you can find interleukins are one of the important carrier molecules, okay, which gives an immunity. And these molecules are secreted by the cells. Okay, so ligand and receptor you can find as the immune molecules in interleukin. So you can find as a best example for signal transduction. In all the cases, only the molecular docking is the common concept, a computational binding process. If you see the molecular docking, majorly you have to remember two steps. One is search algorithm, and the second is the scoring function. You know, we have freewares like Autodock for docking a ligand with a receptor. But if you check with the commercial softwares, generally I prefer docking with Discovery Studio. Okay, Discovery Studio, we have a tool called Discovery Studio Ligand Fit. So I told you Discovery Studio Ligand Fit. This Studio Ligand Fit docks a ligand with a receptor, a ligand with a receptor. But there is also one more module in Discovery Studio that we call it as a libdoc, LIB doc, libdoc. You know, the libdoc is going to do virtual screening. That is, if you want to dock more than 1,000 compounds with a single target receptor, go with libdoc instead of ligand fit. So not only we have just a single molecule, single target docking or multiple molecule, single target docking, we also have modules for docking protein versus protein. So we also have modules in Discovery Studio for docking protein versus protein. Okay. So we have plenty of modules in Discovery Studio. It's a package. One can try anything with a commercial software. Not only that is an advantage with the commercial software, you know, in Autodock, you can dock only a small molecule with a receptor. You can't go with uh, just like that. You can't go with a macro molecule docking with a macro molecule. And even if you want to dock multiple compounds with a receptor, you can't go with a simple Autodock, but we have to go with an Autodock product called PYRX. PYRX is a different software. Okay, generally the Autodock is used for a single ligand, single protein docking. Autodock PYRX is used for multiple molecules, single receptor docking, multiple ligands, single receptor docking. Okay, so but or not only there is a major difference between the commercial software and freeware, also scoring function. In Autodock, we get only one score, that is binding energy score. But whereas in Discovery Studio, you have one module called ligand score. Discovery Studio ligand score. Okay, so in the ligand score, you get almost 8 different scores. You know, each and every score is a combination of two or three physical or chemical properties of the ligand. Okay, we have lick score 1, lick score 2, like that. So the lick score 1 is a combination of the lipophilicity and the entropy. And if you see a second score, the second score is a combination of a pKa and another entropy of ligand and receptor. It depends. Okay, so you have a combination of 2, 3 parameters in each and every score. So through those 8 scores, we can able to identify how exactly your ligand is binding with the receptor, what kind of interactions you have. Okay, so one can use a different search algorithms and through the scoring functions, you can decide which is the best for your project. So ligand database protein, simply if you dock it, you get a ligand is going to bind with the receptor. So the binding can be of a hydrophilic. The ligand can bind either in the hydrophilic site or in the hydrophobic site. The hydrophobic, generally we tell it is a buried sites of the protein. If the ligand is buried inside this protein, which you can't able to see in the naked eye, then it is a hydrophobic binding site. But if your ligand is visible clearly in the outer surface of the protein, then that is a hydrophilic binding site. And what's how a ligand is binding with the disease protein? So generally we do with various computational process. Here you can able to see a ligand. You can able to see a ligand, isn't it? This ligand is binding the receptor, and also the ligand is visible. But you can't tell whether it is a hydrophilic or hydrophobic site because the receptor is in the form of a secondary structure. If only if you convert this protein as a surface receptor, okay, one in a surface receptor, you can able to check whether the ligand is fully buried or not. Then only can tell you is a hydrophobic binding site, or if it is visible in outside, then we can tell it is a hydrophilic binding site. We also have one kind of an algorithm called flood fill algorithm. Discovery Studio uses flood fill algorithm to determine the binding sites. If you have a protein, you simply put a protein in a grid box. In the grid box, you can fill it with water, a medium like water. Wherever the water accumulates, that shows you the hydrophilic binding sites. Okay, you got it. So here the water accumulates somewhere here, so you can find hydrophilic binding sites in the protein. And if you see this classification of docking, okay, not only we can dock a small molecule protein, this is what saying from the beginning. We can also dock protein, the new protein nucleic acid, that is DNA protein, and also small molecule nucleic acid. Okay, this is also a kind of a docking. Molecular complementary in docking. Generally, you might have seen, everyone have seen this kind of a puzzles fitting one piece with another piece, but generally in docking it won't happen like this. In docking, we can find in the form of a chemical complementarity. So more about the chemical complementarity, we can see the description in this picture. So if you see here in this picture, suppose think like this is your receptor. Okay, this is your receptor functional group, and this is your ligand. Okay, if a ligand is interacting, generally the hydrogen bond donor is from a receptor based, and this is a ligand based. So think like it is a drug at the top, and this is protein at the bottom. So the ligand, if it is binding with the disease protein with a single bond, okay, the attraction is minimal when compared to uh, the aromatic interaction. In aromatic interaction, if it is a six benzene ring, you find six hydrogen bonds. If you find this five cycle ring, then you get a five hydrogen bonds. The six hydrogen bonds is more stable when compared to five, and five hydrogen bonds is more stable when compared to the aliphatic bonding, that is a single hydrogen bonding. Okay. Similarly, if you see the electrostatic, so negative terminal versus positive terminal of a ligand and receptor, if it binds, okay, generally you may have a kind of a two bonded, two hydrogen bond kind of interaction. And the hydrophobic is obviously a powerful when compared to electrostatic. Electrostatic is weak when compared to hydrophobic. You can able to see that in hydrophobic you find nearly four kind of a bonds. So you find good kind of an attraction in hydrophobic when compared to the electrostatic. This is what you can understand. Okay, now let us take you to the next slide. That is talking in hydrophobic binding site. So generally the hydrophilic binding site looks like this. So an aliphatic bonding is a single bond. But the hydrophobic context binding is something buried inside. Okay, you can't able to see in the naked eye if you rotate the visualizer. The drug is present somewhere inside the protein. Then whatever you find the negative and positive attractions in the hydrophobic interactions give a kind of electrostatic and salt bridges. If salt bridges are formed, these are covalent interactions. Whatever I told, even hydrogen bond interactions are non-covalent interactions. Covalent interactions are very strong. So the drug protein binding is strong, and so the drug is going to reside more time. The drug is going to reside more time. The residence time is more when compared to this kind of interaction, the hydrogen bond kind of interactions. So whenever you find a drug dosage is taken only once in a day, and even if a drug dosage is reduced, then obviously it is a kind of a hydrophobic interaction is happening. And if you take paracetamol, for example, if you take a paracetamol, we take 3 doses a day, isn't it? If you are a heavy male or female, that is 75 kg or above, the doctors, the clinicians prescribe 650 mg, you have to take for three doses, morning, afternoon, and night. Okay, then you will be taking accordingly. But think about a situation that a person is having a upset in bowels, that is facing a kind of a loose motion. Then how? What kind of a drug we take? Loperamide, isn't it? Loperamide. It is marketed in different names. Generally, you take a loperamide tablets. Whatever loperamide you take, it's very less in size. We are not going to take even 500 mg, but just a 50 mg is sufficient. And you know that 50 mg is going to target a large intestine. If you stretch your intestine, it is in meters, okay? Okay, and up to the so whatever size, okay, it doesn't matter. Only how your drug is going to target in the binding sites. That's the most important. So in the later case, the loperamide, you can find the drug is binding the hydrophobic sites, not like the hydrophilic sites like paracetamol. So when you're taking multiple doses and a large dose when compared to loperamide. Now, energy dictates molecular association. So if you see how a ligand is binding to the receptor, see whatever you are seeing, this small thing is a ligand, the light blue, and something like a dark blue is a receptor. If you see this binding is not perfect, this binding is also not perfect, but this binding is perfectly bound with a maximum surface. The ligand is maximum bind with the receptor. So if this is the best binding, so you get a kind of a negative maximum energy. So this negative maximum energy we consider as the base binding energy of a ligand at the receptor. The remaining binding role we can't find as the best one. And if you see the types of docking, okay, one type of docking is not possible. If you check with rigid ligand, rigid protein, this docking is possible. Flexible ligand, rigid protein, this docking is also possible. Rigid ligand, flexible protein, which is not possible. One type of docking is not possible in the softwares. Okay, that is this is possible. This is not possible. The only docking which is not possible is rigid ligand, flexible protein. For this alone, we don't have any softwares at present. The softwares we have the flexible. Even most of the commercial softwares uses this kind of a docking. Okay, most of the commercial softwares uses the flexible ligand and flexible protein docking. Okay, so up to present we don't have this docking alone. Protein ligand docking. We have plenty of softwares, nearly 500 softwares will be there, but the offline softwares will be few. Okay, and generally many scientists try to do Autodoc software. Okay, so the Autodoc you can download from scripts.edu. Don't download in any other websites. This is the home page to download the Autodoc software. So I think many ask about to proceed for molecular docking using Autodoc. So during the mini project discussion, I'll be explaining you how to use Autodoc and Autodoc PYRX. Okay, I think that gives you a wide range of applications and you can find use in many projects. Okay, but if you don't want this and if you want some other applications, then you can ask me, then I can take you accordingly. Okay, so Autodoc is the freeware. If time permits, I will be teaching you that. Okay, and protein talking generally I go with HADOC, FireDoc, HexDoc. These are the general doc softwares we can use, but we can also try to use these softwares what I mentioned. Desolvation energies. Okay, this is a concept in detail. Entropic effects. There's a lot to know actually. For molecular talking, we have textbooks. Okay, for homology for modeling and simulation, I prefer a textbook called Anand Solomon, an Indian author textbook. That is a small, a thin textbook, but you find major concepts like how I am showing you the slides. Of course, these are basics of drug design. Okay, entropic effects and all these things. So we'll be discussing that. Okay, now we let me take you to the most important thing. We should know at least the chemistry and what's happening inside. Or if you see the molecular talking, a general ligand is going to bind with the protein, and a ligand if binds with the protein, okay, you find a complex, isn't it? So the energy binding energy is generally calculated. Whatever del G here we mean a change in Gibbs free energy. The change in Gibbs free energy is what you call calculate as a binding energy even in autodom. So how this is calculated? The energy of complex, okay, whatever you formed here, complex minus E protein minus E ligand. This is the formula for binding Gibbs binding energy. And you know already from thermodynamics that the change in Gibbs energy is nothing but the difference of change in enthalpy to the product of temperature and change in entropy. Okay, del G equals del H minus T del S. So this is the formula. And of course, so the del G binding, which is ultimately we found from the docking software, is calculated like this: complex minus that is product minus the energy of reactants. Okay. Now I am going to take you to the next slide. There are different formulas for calculation of energy. This is one more formula which is taking care of bonded interactions or non-bonded interactions. You remember when I explained you related to the simulation that in the molecular modeling that generally we find bonded interaction, sum of bonded and non-bonded interaction is what the you take it for calculation of potential energy. So similarly here also. So generally, if you see the non-bonding energy, van der Waals energy, and electrostatic, the remaining hydrogen bond, torsional, desolvation, everything we can also take in bonded interactions. Molecular dynamics. Remember this picture? I explained you bonded interaction and non-bonded interaction. That is bonded energy and non-bonded energy sum up to form a total energy. The softwares for working with molecular dynamic simulation is GROMACS, AMBER, NAMT, and DESMOD. So one should know what is the major difference between molecular docking and molecular dynamic simulation. So molecular docking, still you find same thing like a ligand is binding with the receptor. Even in molecular dynamic simulation, still you study the same, but there is a slight difference. Here we are telling about dynamics. So here, whatever ligand is going to dock with a target receptor, you study with respect to a period of time. You got it? In molecular dynamic simulation, okay, we generally dock a ligand with a receptor that you take it as an input. That is a docked ligand receptor complex, you take it as an input and you study over a period of time through various analyses like RMSD, RMSF, etc. So why we have to do over a period of time? You know, when a ligand, when you take a drug through the oral route, it can be even a paracetamol. One should know how much time a paracetamol is going to reside in our blood. You know, it takes the residence time is just 8 hours. So one day the doctor is prescribing to take 3 doses a day. So for the 24 hours, I find that the drug will be circulating in our blood. You got it. So the residence time is most important. So how we can able to find like how a simulation is happening in our body, how a paracetamol is going to assimilate in our body. The similar thing we are estimating in the computational techniques in the software itself. The software we use is the MD simulation software like this: GROMACS, AMBER, NAMD, and TESMOD. There are different search algorithms I told you earlier for which distinguishes a freeware with the commercial softwares. Also, we have scoring functions. Okay, so there are different methods of talking. The most important one I can take. Okay, so okay, the fragmentation docking method. Okay, like if you have this as a drug, okay, and you can also fragment it. You can break wherever you want. Okay, and whatever fragments you have, you can able to link with some other fragments instead of having this entire drug. If I link a fragment like this, and if this drug is going to give a better efficacy for a disease, okay, then I can take this as a granted drug. So when compared to the normal drug, I just initially fragment it and I just linking one fragment with another fragment, and whatever I form as an active pharmaceutical ingredient, if this is going to give a kind of a remedy for a specific disease treatment, uh, and showed better when compared to the already existing drug, then I can take this drug as granted. So for this, I can use fragmentation docking method. And we have incremental based model methods, placement, and these are some of the algorithms. Okay, so one should know what exactly we are using. Yeah, so this in this slide you can see what is virtual screening. As I already told, if you go for a docking a single ligand with a receptor, we can't come up with a solution. If and if you take multiple compounds and you dock with the receptor, then only you can come to know which is the best drug. This can be determined only through the visual screening. So picking a best drug from a multiple drugs. Lead hopping. This is similar to the fragmentation based method. The fragmentation based screening, the other name is lead hopping. So you fragment, you just cut into the many fragments and you can attach with the linkers with the desired fragments. So initially you have the fragment like this, but finally you have the fragment like this. So if you see this, the drug structure, this and this is different. But if this reassembled fragments give a better efficacy, then you can take this as a base drug. You ignore this drug, already drug in the market. So lead hopping can give you best results and increase high throughput screening rates. So for this, I can give an example like what we tried in IGEM project. In IGEM project, International Genetically Engineered Machines competition, usually conducted by Massachusetts Institute of Technology, USA. Okay, the SVC students have participated around four. On all the four competitions, they got awards, silver and bronze awards. And if you see this kind of a innovation, what they tried in IGEM is they the in a project they want to avoid spoilage of milk. For avoiding spoilage of milk, if we mix a peptide, okay, the shelf life of milk is increased. For example, if you purchase Avin or Nandini, Avin from Tamil Nadu or Nandini from Karnataka, the shelf life of a purchased milk packet is just 2 days, isn't it? And even based on how you keep in refrigeration. Okay. The maximum shelf life they ask you to immediately have to keep it in refrigerator. And if your refrigerator is not working, if you have power cut problems, then obviously you find the milk spoilage. But if I want to keep shelf life more than 2 days, that is up to 3 months, up to 4 months. You have heard something about ultra packed milk. Okay, similar to that, even the students have tried by mixing peptide in the milk. If they mix the peptide, it can serve like an ultra packed milk. Okay, the shelf life can be increased to 3 months. So for that, we came to know that we should develop a 9 amino acid peptide, sorry, 9 amino acid peptide, and the last two amino acid, that is eighth position amino acid and ninth amino acid position, it should be a tryptophan. If the eighth and ninth are tryptophan, the remaining 1 to 7 can be a random of 20 amino acids. The 20 amino acid single letter characters, isn't it? The first 7 characters should be a randomized 20 letters amino acid. So you get thousands of compounds. Whatever thousands, it's not a compound, thousands of peptides. Whatever the thousands of 9 amino acid peptides you got, we have to screen it like a virtual screening that we tried with high throughput docking. Okay, if you check with the small molecules, you can tell it is a screening, but this is a kind of a peptides we developed, okay, based on the randomization process. And now we go for a high throughput docking and we find the best best peptide for avoiding spoilage of milk. Okay, so this kind of a project we tried, and of course, this is a product. And for computational process, I helped them. Of course, they got silver medal. They appreciated just because of computational method, they got a silver medal. But even they tried with genetic uh, engineered principles. They tried with that also. Okay, now let me take you to the reverse talking. The reverse talking is just quite opposite of talking. Generally, if you have a ligand and receptor, then you can talk, isn't it? Here, initially, in the acid, that is a reverse talking software. Okay, it asks you to choose a 3D structure compound file. So if you have downloaded a compound file, can be a curcumin in the form of curcumin.htf, upload that file, and there are series of receptors, like classes of protein target receptors. Okay, so if you want to check with a specific target, you check it, or simply you have to click submit. Then your drug is going to bind with which major receptors that you can see. If you see here, okay, for a drug called vitronide A, there is a plant-based compound. I got maximum targets. It is maximum binding with oxidoreductase receptors. Next to oxidoreductase, transferase, hydrolysis. Okay, the hydrolysis or enzymes, proteases, lipases, all these things. So this ligand is binding with the third best is hydrolysis, but number one is oxidoreductases. Oxidoreductases helps in redox reactions, helps in treatment of cancers. Okay, these target receptors. So likewise, you can able to check. Okay, like with a drug molecule, you can check what are the best binding target receptors. Okay, so this is something about my institution. Uh, this is not yet updated, but we have a few more concepts like for modeling, PSAR. So many concepts are there, and even I did teach you practical skills on talking, virtual screening. So we all see during the mini project discussion, if you permit. Okay, any doubts at present? Sorry, any doubts you have at present? So till now we are seeing only the theory concept. Theory itself we have a lot. Okay, so if you have any doubts, okay, or if only if you study, if you have a doubts, you accumulate it and ask in the next session. Okay, so thank you for the patience and interactions, participants. Have a great day to you all.